Today we will continue our discussion about this cortical plate development gets affected when the calnexin is not present in the cells. So this figure discuss about the results of the experiment on the mouse model where calnexin has been knocked out. So we will discuss how this calnexin knockout affect this cortical plate development. In our previous video, we have understand the graphical abstract of this paper, that what are the main results that they have find out. So, first thing that they did is they analyzed the different levels of this cortical plate development, which has been characterized into this 1, 2 to 4, 5, 6 in with the help of this DAPI staining CUX1, CTIP2, and this is the combined images. So when the calnexin are present, they can see that signals for both the CUX1 and CTIP2 are normal. However, when these calnexin are absent, they can see the reduction in the signals for this CTIP2, and also some of the reduction in this CUX1. And when they better did the quantification analysis, they are able to find out that the signals for this layer 2 to 4 are significantly lower in the case of this calnexin, in the case of mice where calnexin has been knocked out, compared to the case where calnexin is not been knocked out. However, for level 5 and level 6, the difference is not statistically significant. So they focus on this layer 2 to 4. So after validating that there is a change in this cortical plate development at layer 2 to 4, they decided to check whether there is an effect on this Trkb or phosphorylated Trkb. And when they check in the mice model that is calnexin is present, they can see that high signals for the phosphorylated Trkb. However, when these calnexin are absent, they can see some reductions in the signals, and this is the negative control where this phosphorylated TRK is not present, so they don't see any kind of signals. And when they do the immunoprecipitant assay against this TRKB, they can find out that in the case where calnexin is present, they can see both the TRKB. One thing that we need to know that phosphorylated TRKB has a molecular weight around 100 kilodaltons, whereas cell surface TRKB has a molecular weight of around 130 kilodalton. And they attributed this difference could be due to the difference in the glycosylation of the phosphorylated TRKB and the cell surface TRKB. So in the case of this phosphorylated TRKB, they can see that more phosphorylated TRKB are present compared to the normal cell surface TRKB when the calnexin is present within the samples. And in the case where the calnexin is absent, they can see the similar TRKB. So let me correct myself. So here in the top panel of this, in the top figure of this panel D, this talks about only the N-terminal TRKB. So here they can see that N-terminal TRKB are present in both sides. However, the phosphorylated TRKB, which they talked about in the bottom panel, it's more in the calnexin knockout compared to the case of this calnexin positive, where calnexin are present. This could be because of the lack of movement of the phosphorylated TRKB to the cell surface and accumulation of more phosphorylated TRKB within the endoplasmic reticulum or in the cells. That's the reason there is a high concentration of phosphorylated TRKB, because it is being cells are being overcrowded by this TRKB due to the absence of calnexin that helps in the proper folding, at the same time in the transport of this phosphorylated TRKB to the cell surface. And these things they have validated here using this anti-TRKB antibodies, and this time what they did is they used these IgG antibodies, in which case they can see almost no signals. This is act as a control, and whereas when they use these Trkb antibodies, they can see the high amount of this Trkb. Both they can see the high amount of Trkb. That is at around 100 kilodaltons and also at around 130 kilodaltons. And when they check the phosphorylated TRKB to the TRKB ratios, they can see the phosphorylated TRKBs are high, uh, and the ratio is high in the case of this calnexin is knockout when compared to the case where calnexins are present. And this knockout between and how they differentiate the phosphorylated TRKB and normal TRKB that I have just mentioned is that phosphorylated TRKB has around molecular weight of 100 kilodaltons because of the difference in the glycosylation profiles compared to the case of normal TRKB, which is a molecular weight of around 130 kilodaltons that are expressed on the cell surface. So if we check the ratios in the case of this calnexin knockout compared to the case of their calnexin are present, we can see that the ratio is higher. It indicates that phosphorylated TRK is higher in the case of calnexin knockout because of the overproduction of this phosphorylated TRKB within the cells and lack of transport of this CRKB, phosphorylated TRK onto the cell surface. And after that, since there is a change in the glycosylation profile, they decided to check how much is the immature glycans are there and how much are the N glycans are there. So in order to check the immature glycans in both the phosphorylated and normal TRKB, they use ENDOH, and they can see the shift in the Western blot, both or shift in the molecular weight, both for this phosphorylated and the cell surface TRKB. And same things they have observed when the calnexin is knocked out, they can shift in the phosphorylated TRKB and the cell surface TRKB. And when they use PNGSF, they can see further shift. It indicates that it has the N-glycan structures also, and these shifts are also visible in the case of these calnexin knockout samples. So this indicates that both the phosphorylated TRKB and TRKB has immature and N-glycans in it. And thereafter, they check the effect. They check the effect of this Calnexin knockout in the case of this N-terminal and this C-terminals TRKB. And in the case of N-terminal, both in the low exposure and high exposure, they can see almost negligible difference. Both are similar. Whereas when they check it for this C-terminal, what they can see is that for the C-terminal, they can see some changes compared to the case of where these calnexin knockout are present and calnexin knockout are absent. And this is the HSP90 is the control for the Western blot. And so this is the quantitative analysis that they have done of these Western blots. That for when the N terminals for the N terminals at 130 kilodaltons, they can see some of the significant changes. However, in this and for this 100 kilodaltons, the TRKB N terminals, they can also see they can't see any significant changes. So this indicates that, so let me correct myself, for 100 kilodaltons, since it represents the phosphorylated TRKB, they can not see the significant changes. However, for this 130 kilodalton, that is cell surface TRKB, they can see the significant changes. So this indicates that the cell surface TRKB has been reduced in the case of this calnexin knockout. However, the cell surface CRKB of N terminal reduced in the case of this Calnexin knockout. However, there is not much impact on this phosphorylated N terminal CRKB, TRKB. However, when they check it for this pen TRK, they can pen TRK or C terminals, they can find out that there is a difference. There is a difference in the case of this pen K TRKB that is non-phosphorylated. Both in the term in the case of this Calnexin knockout, it is reduced, significantly reduced, statistically reduced compared to the case of this where the Calnexins are present. So thereafter, all of this with comes to a conclusion that there is not much impact of this Calnexin on this N-terminal part of this TRKB, phosphorylation of this N-terminal part of this TRKB. However, the non-phosphorylated part, or the part that has been expressed on the cell surface, is got affected, and same thing they have seen for the C terminals. So this indicates that TRKB transport of TRKB has been role, or we can say that Calnexin plays a role towards transport of CRKB, as the absence of Calnexin will cause reduction of this TRKB both in N terminal and C terminals on the expression on the cell surface. Both in terms of N terminal and C terminals of this TRKB are low. It indicates that TRKB could be expressed low on the cell surface. Not like this. Since the signals for both the N terminals and C terminal TRKBs are low, non-phosphorylated, this indicates the non-phosphorylated TRKB, which are usually expressed on the cell surface, are low, because the phosphorylated TRKBs are there is not much phosphorylated TRKBs and terminals, not much difference has been observed. However, they have not. However, for this C terminals, they have not shown the results for the C terminals, but it looks like that C terminals phosphorylations also C terminals phosphorylation seems to be increased compared to the case of this non-phosphorylated TRKB. So this further indicates that P terminal phosphorylations are why the phosphorylations are overcrowded in the case of Calnexin knockout. Next, they also check whether the signals are positive only for the TRKB or TRKA and C, and when they knock out this NTRK2, and they cannot see the signals for this TRKB. So this indicates that the Western blots, they gives the signals only for this TRKB, and it does not have any TRKA or TRKC, as this NTRK2 negative knockouts does not have TRKB, so we do not see any signals. It has the TRKA and TRKC, but it does not give any signals. So this indicates that Western blots, it gives the signals only for this TRKB, both in C terminal and N terminals. So I hope you got some idea about how this mice model results are being analyzed to come to a point that Calnexin knockout plays a role towards this TRKB phosphorylation, and that is known in the literature to be correlated with this cortical plate development, thereby affects the brain high functions. So that's all for this video, and if you need to understand more about this experiment in detail, you can go through the reference that has been provided in the description. Thank you for your time.